Hello everybody, welcome to this week's video. This week we're going to be talking about the plasma membrane and its role in homeostasis. So the plasma membrane surrounds the entire perimeter of a cell. It contains a phospholipid bilayer that regulates the movement of molecules inside and outside of the cell. The membrane consists of hydrophilic phosphate heads and hydrophobic lipid tails. The transport across the plasma membrane depends on the concentration of gradients. Differences in concentration between inside and outside of the cell will determine where a molecule will move, and if the molecule can move or not. Gradients are essential because they determine movement and they keep living separate from non-living. Life relies on cell activity, so materials moving in and out of cells will solidify or expand their composition and thus increase activity and life. These gradients must be maintained in order to keep areas in a state of homeostasis. This will inevitably keep areas different from one another. Through the maintenance, molecules are actually able to move and allow certain processes to work. However, our bodies must work against the gradient in order to maintain it, which costs energy. We'll speak a little more on this later. Now let's talk about some of the components of the plasma membrane. The membrane contains proteins, cholesterol, glycoproteins, which are carbohydrates attached to protein, and glycolipids, which are carbohydrates attached to lipids. These components are all essential to its function. Let's talk about the proteins a little bit. We have two types of proteins attached to the membrane. First, we have peripheral membrane proteins. These are temporary membrane attachments that exist just on the outside of the membrane and are used for cell coordination and communication, or even as enzymes. Next, we have integral membrane proteins. These are permanent attachments that integrate or connect all the way throughout the membrane. They have hydrophobic regions and hydrophilic ones to tend to both sides, both layers of the plasma membrane, and they're used for transportation of hydrophilic molecules across a membrane, or could also even be used as cell receptors, which receive signals from other cells or even their own. There are also different mechanisms in which molecules can be transported. So large molecules and particles are transported by membrane-bound vesicles. So when a large molecule or particle wants to go inside of a cell, it's called endocytosis. This is done in three separate ways. First off, we have phagocytosis. This is specific to larger particles and is also referred to as cell eating because a vacuole is formed between the plasma membrane and the particle, and that forms this sort of circular vacuole that is then allowed to enter the inside of the cell. We then have pinocytosis, which is also referred to as cell drinking. That's because these are concerned with smaller particles. A vesicle is formed between the membrane and these small particles and are easily able to just be transported into the cell. Finally, we have receptor-mediated endocytosis. This is very selective, as an external receptor will bind to a specific ligand, which is basically a chemical messenger released by a cell. This will form a vesicle and then enter the cell. So that's into the cell. What about outside of the cell, which is known as exocytosis? Materials here are going to be packaged the same way they are in endocytosis, into their own vesicle, but then when they actually want to exit, the membrane of the vesicle will mesh with that of the plasma membrane, and then the contents will be released, exocyted out of the cell. So now let's talk a bit more about the different ways molecules can cross the membrane. Let's start with smaller molecules. These small molecules, the hydrophobic ones, cross the membrane extremely easily by themselves in a process known as passive transport or diffusion. This is when a molecule moves along its own concentration or electrical gradient. Examples of this include oxygen and carbon dioxide, and even though it's polar, sometimes water can even do this. Even after the cell has reached equilibrium, there is still molecule movement. Even though there's no net flux, molecules will still be moving, increasing and maintaining cell activity. How is the rate of diffusion impacted? Rate of diffusion will be increased with several different factors. First off, a stronger gradient, higher temperature, decreased membrane thickness, greater surface area, less distance traveled across the membrane, and a higher membrane permeability. These will all increase the rate of diffusion. What actually increases higher membrane permeability is a smaller molecule size and higher lipid solubility, considering that the inside of the plasma membrane is hydrophobic. This makes perfect sense. Now let's talk about very polar or larger molecules. How will they get across the cell? Because obviously they cannot just easily diffuse across. These will be through protein channels and carriers. A protein channel may either be open or closed. When it's open, otherwise known as a leak channel, molecules can pass through without any sort of bonding, sort of like a car driving through a tunnel. When they're closed, however, bonds must be created with carrier proteins, which will then transport the molecules across. These carriers, however, have a limited capacity. This could be maxed out by more bound molecules, and it limits the amount that can go across. When there's a big or stronger gradient present, the carriers are slowed down. Channels are further used for diffusion of ions. These channels are either open or voltage gated. When bound to the gated channel, the charge on the ion will control the opening and it will transport across. Now for the second to last form of transport, we have active transport. Active transport is the movement of particles against a concentration gradient. This uses a carrier protein, effectively a pump to push molecules from low to high concentration. A practical example of this is a sodium-potassium pump used in an action potential. This pump uses energy, which is ATP, to push 3 sodium ions out and 2 potassium ions back into the cell to restore the cell concentration. Now, because of the use of ATP, this is also an example of primary active transport. This is where energy is directly used to move a molecule against the concentration gradient, or electrical gradient. Finally, we have secondary active transport. This is where the energy released from the diffusion of the first molecule through primary active transport is used to move a second molecule against its own gradient. So the ATP used to move the molecule 1 creates an electrical field that propels a second molecule. These are done through 3 different transporters: a uniporter, symporter, and antiporter. A uniporter carries 1 molecule or ion, a symporter carries 2 different molecules or ions, both in the same direction, and an antiporter carries 2 different molecules or ions, but in different directions. The sodium-glucose pump is a prime example of secondary active transport. Thank you guys for listening today. I'll see you next week.